Hi there, my name is Scott Phillips. I'm the president and founder of Starfish Medical. In this video, you'll learn a little about our bioservices offering. It's a really interesting area. We've been doing it for a few years. We've managed to attract some wonderful people to it. And where we're really focused is on diagnostic assay technologies, advanced cellular therapies, on microfluidics. We help companies figure out their product, the whole value proposition, getting that dialed in. We're really the partner for the lean startup journey for these companies. And then we help them get the products ready for manufacturing, prove that they're scalable so that they're really ready to scale up into the market. I'm sure you'll really enjoy these videos. Thank you.
Hi there, my name is Allan. I'm the functional manager for the bioservices group here at Starfish Medical. One of my areas of expertise, if you will, is biofilm growth. And as a researcher, I was really interested in how biofilms of microorganisms grow on implant medical devices. And that's an interesting concept because it's fine to grow microorganisms in a culture flask, but in order to grow them on a medical device, you need a surrogate and a way to screen them in a high throughput way. And so early on in my career, I was working on a device. It's fairly simple by today's standards and the things that we do here at Starfish, but here's a very simple, in fact, this is my first medical device that I developed. And what we would do is grow the biofilm on these little pegs here, expose them to different reagents, and see how well the device or how well the reagents performed. Now that's very interesting. From a development standpoint, we're just basically doing things in the lab, got some injection molders, made some parts, put these things together, and designed this product basically just to do research work. And that's fascinating when you're doing the research. However, as we were able to make some pretty exciting advancements with this technology, we quickly found it had a very specific application. That was screening antibiotic combinations for use in very serious disease, in genetic disease called cystic fibrosis, where you can't clear the microorganisms that grow in your lungs. It was a powerful screening tool to do that. The problem was we developed this in the lab with just engineering it to serve the function, that's growing microorganisms. We didn't do it under an ISO 13485 environment for medical device design. And so what the ramifications of that was, so it's a very straightforward, very simple product by today's standards and what we do here at Starfish. It took a very long time to get from that product development phase to a product phase because we did not design under ISO 13485. We had to backtrack and do that, and that added many more years to the development process. So if I can take a concrete example to show you about why this is so important, here's a very tangible example right there.
Hi, my name is Lorenzo Gutierrez. I'm the Microfluidic Manager and a Bioprocess Engineer at Starfish Medical. I have been with product development for over 30 years, and for the last 10 years, I've been working on point-of-care devices for a variety of disease panels. In my line of work, we usually do immunoassay, molecular, or cell-based assay. We work on projects that require cell culture, characterization, sorting and detection, and reagent formulation, including biomaterial products, to support our clients. We develop and build associated instruments and fixtures, as well as prototyping and production processes. For biologists and bioprocess engineers like me, we spend most of our time doing cell culture, process optimization, and in some projects, small-scale manufacturing. Utilizing microfluidic technology for biomanufacturing is an advancement in developing or testing new biomolecules or biologics for a variety of medical uses and therapies. One of the promising microfluidic applications for biomanufacturing is the production of cell line in a parallel system. Considering the ability of microfluidics to manage small volumes at the level of a single cell. I was really fascinated with the many studies that demonstrated the idea that we can use microfluidic to mimic body tissue or organ, and hopefully manufacture it in the future. Also, the ability of microfluidic to produce tiny droplets and nano or microsphere, as well as the ability to encapsulate, will open up new ways to produce an efficient drug delivery system that could control its release in the body. And finally, the production of tiny encapsulated bubbles using microfluidic can be used for preserving cells. This ability could be very useful for 3D bioprinting of tissues and organs in the future. Microfluidic technologies and devices being designed and built today will support and improve biomanufacturing.
Hi, my name is Mark Derlick. I'm the Director of Program Design. My role within Starfish is to try to understand technologies and develop programs that enable client success. As we think about bioservices and biomanufacturing, one of the things that often is required is the delivery of those therapeutics to the body. One of the projects that Starfish was involved in was the delivery of stem cells to the ventral horn of the spine. Our involvement in that project was from very early stage, trying to conceptualize what and how the device was going to function and deliver stem cells to the targeted region. It involved some early stage design, development, workflow development, early stage prototypes that were used in preclinical porcine studies to try to determine how and what is working and what is not. Through a series of iterative design prototypes, we also created a design history file. The system itself consisted of two primary components. One of them was a stereotaxic positioning system, which anchored itself on the surrounding tissue of the spine. And the other element of the system was a cannula. And that cannula was intended to take the stem cells and target them to a specific depth within the spine. After we had decided what the device was going to require and need in the general design, we executed upon that design and took the various elements through the appropriate verification and validation protocols. And in this instance, it was processing of the reusable components to determine that they could be sterilized and reprocessed. Then we took all the outputs from the verification and validation activities and compiled them into a design history file that was provided to the FDA as a part of a device and drug filing that enabled clinical studies. To this day, Starfish continues to support these clinical studies with the manufacture of small volume cannulas and stereotaxic devices. As stem cell therapies are not fully in the market now, we understand that there will be clinical trial support that's required. Starfish continues to support the manufacturing of those devices as well in support of those clinical studies.
My name is Dr. Stephanie Willarth, and I am a full professor and Canada Research Chair in Biomedical Engineering at the University of Victoria, and I'm also a Bioservices Advisor here at Starfish Medical. Today I wanted to talk a bit about the current biomanufacturing ecosystem here both in British Columbia as well as in Canada, as it's been quite productive in recent years. Even throughout the COVID-19 pandemic, we've had some of our Canadian-based technologies really become internationally adopted, and partially that's due to the quick acceleration of taking some of these technologies from the prototype stage all the way through to clinical translation. And I think here in Canada, there's particularly some exciting reasons why it's more accessible to translate these technologies for clinical applications, especially in some of the up-and-coming fields such as cell therapy. Here in British Columbia, we have two first-in-class clinical trials currently. One is local here in Victoria with CAR-T cancer stem cell therapy being developed at the British Columbia Cancer Agency. And across the water at Vancouver, Tim Kiefer's group at UBC in collaboration with Biosite have recently published some groundbreaking data in cell stem cell about using cell therapies to reverse the effects of type 1 diabetes. Starfish Medical has some really interesting expertise in being able to help companies access these types of technologies. For example, their experience with medical devices has led them to make systems that enable checking the quality of the different cells that are necessary for cell therapies, innovative systems for separating different types of cells, as well as developing bioreactors to scale up the amount of cells needed for these different therapeutic applications. I think one of the strengths is being able to take some of these technologies from the prototype stage and enabling them to be translated all the way through for clinical applications, as I've previously mentioned with some of the recent examples that have been success stories here in British Columbia.
I'm Joris van der Heijden. I'm the program manager in the Bioservices group at Starfish Medical. And we've made a lot of medical devices, but we actually also in the Bioservices group, make a lot of tools that enable other companies to make their drugs for clinical trials, especially most recently when you've seen the move from small molecules to biologics. They come with their own challenges, and especially there we can do a lot by make specialized equipment for releasing their drug products on market. If the engineers that make the equipment work together with the scientists and the engineers that are making the drug, then we can really truly make magic happen. And the immediate examples that come to mind are things like personalized antibody treatments, personalized cell therapies, where as a cancer patient, you go into the hospital, you get your specific cancer genotyped or sequenced, and based on your sequence and your personal cancer profile, you'll get your personal therapy. And right now, what's still standing in between the theoretical reality of that is that we need to find better manufacturing solutions to really bring that type of therapy to the patient. So really what we can do at Starfish is make the specific equipment that would enable personalized medicine, for example, something like antibody treatment. And if you think about that, let's say a cancer patient can go to the hospital to get the profile of their specific tumor through sequencing or proteomics or any other screening method. Now we have a particular profile, and there is already the technology to make a therapy related to that particular profile. But Starfish can now help to make that therapy specifically for that particular patient in small volume and with very rapid release and testing, and then that patient can be helped in a much more effective manner in a short period of time.
If you'd like to know more about Starfish, we've been doing this for 20 years. We've developed hundreds of devices for companies all over the industry, and we've helped many founders become very successful and have big clinical impacts. That's what we really love for. So enjoy the content. If you'd like to talk to us about your project, we'd be happy to talk. Thank you.